Good morning, students. As we have already done about the zwitter ion, now we'll talk about the structure of proteins. Now the structure of proteins, which is very important, as we know that the amino acids, when they join together, they make proteins. So what kind of bonding do they have when the one amino acid joins with another amino acid? The bonding between the two amino acids is called peptide bond, which is very important, and it comes in the exam also. Or it is also called peptide linkage because they are linking with each other. So amino or amide formed between COOH यानी कि carboxylic group and NH2 group. So here one amino acid, this is one amino acid and this is second amino acid. So when one amino acid joins together with the second amino acid, what kind of linkage do they have? C double bond O NH. There is a release of water. There is a release of water means one H is gone from here and OH is gone from here. So H2O. H2O is gone. So the linkage is C double bond O bonding, hydrogen bonding, and H is already gone. So one H is left. So this kind of linkage between the two amino acid is called peptide linkage. So it is between the glycol and alanine. So that's why it is called glycol alanine. So this is glycol and this is alanine. These are two amino acids. When there is a release of water I have shown you this OH is gone from here, H is gone from here. So CO can be written as C double bond O, NH which is already written here, and the linkage between them is called peptide linkage or the peptide bond between the two amino acids. It is called peptide bond or peptide linkage. I hope the concept is clear to everyone now. Now we'll talk about the molecular structure of the proteins. We'll talk about the molecular structure of the proteins now. On the basis of the molecular shape, they are fibrous, and they are globular. Now listen to me very carefully. Polypeptide chains run parallel, means they run parallel with each other, held together by which bonding? Hydrogen you know very well, but there is a disulfide bonds also. When the proteins work together and there is a linkage of the hydrogen bonds as well as disulfide bonds, they make fibrous proteins. Fiber-like structure is present in between them and they are insoluble in water, which is very important, you need to learn. And the examples are always asked in the entrance exam also and in the paper when you have to give the final exams. Example is keratin and myosin. Now where do you find keratin? In your hair, wool and cell. Where do you find myosin? That you find in the muscles of the body. Second one is the globular protein. When the chains of polypeptide, they coil around to give a spherical shape. You know spherical shape? That spherical shape, when it is formed, that's why the name is globular is coming, right? It is soluble in water. What is the difference? It is soluble in water, but it is insoluble in water, which is the fibrous ones. And what are the examples? It is insulin, which maintains the sugar in the body, and albumin. So these two examples are very important. You need to learn. It can be asked in the exam that what is the difference between fibrous and the globular proteins, or on the shape of the molecular shape, how will you differentiate between proteins, or what are the different types of proteins on the basis of molecular shape. So the next one which is coming up is now structure and shape of proteins. Now the structure and the shape of the proteins. Now they are primary, secondary, tertiary and quaternary. Right? Shape of the proteins are primary, secondary, tertiary and quaternary. If, listen to me very carefully, which is very important, you need to understand. Proteins may have one or more polypeptide chains, which I have already mentioned here. They can be having one or more polypeptide chains. If they have one or more polypeptide chains, each polypeptide in a protein has amino acids linked with each other in a specific sequence. I am stressing on a word specific sequence. Then the sequence of the amino acid is called primary amino acid. What do we call it? Primary protein or amino acid, right? Secondary, the shape in which a long polypeptide chain exists. These are the main words which I am highlighting. You need to write in the answer sheets, otherwise you will not get the marks. And there are two types, alpha helix and beta braided sheets. Secondary protein is further divided into alpha helix and beta plated sheets. It is always asked in the exam that what is alpha helix? It is one of the most common ways in which a polypeptide chains forms all possible hydrogen by twisting into a right-handed screw with an NH group of each amino acid residue hydrogen bonded to the keto group of an adjacent turn into a helix. So that is called alpha helix structure. But the beta plated structure, all peptide chains are stretched out to nearly maximum extension, and then that can be laid side by side, which are held together by the intermolecular hydrogen bonds. Intermolecular hydrogen bonds, that is called beta plated sheets, which is very, very important you to remember. Now we go to the tertiary structure. Tertiary structure represents overall bonding of the polypeptides, and they are in the more coiled form, right? When primary and the secondary structure coil together, they form the tertiary structure. Now the last one, which is the quaternary structure, which is very important. It is made up of 2 or more polypeptide chains referred to as subunits. The subshell arrangement of these subunits with respect to each other is called quaternary structure. So what we have read if it is a long chain sequence, normal sequence in a proper line, it is called primary. If it is in a coiled form, it is called secondary. If it is more thickened and more coiled, it is called tertiary. If all of them coiled together but you don't know what exactly the shape is, it is thin but it is highly coiled, it is called quaternary structure. So with this we have come to the end of this structures, which is we have talked about the structure of the proteins, right? In the next lecture I will talk about the denaturation of proteins. Bye bye, take care.